bacteria. Prokaryotic organisms that are haploid. Bacteria are a very unique group of organisms. They are considered to be the oldest type of organism around. We know they date back 3.5 billion years, and theoretically they are the basis for everything that came afterwards. When we look at a prokaryotic cell, we see that there's not a whole lot here. This particular image shows the outer part. It says it could have a capsule in here, and we'll talk about that. Inside of that, you've got a cell wall. The cell wall is a little bit different. It's got a plasmid membrane, which is the same sort of plasmic membrane that you find in everything. You've got a nucleoid, which is made out of DNA. Now notice, it doesn't call it a nucleus. There's no membrane around the outside of it. It's got cytoplasm, which is the fluid on the inside. It has got ribosomes, but the ribosomes here are 70S ribosomes. They're a little bit different than the ribosomes than you or I would have. And then it's got little things on the outside that are called pilus, and these are little things that allow them to sense what is going on around them or know if something is coming in contact with them. That's your prokaryotic cell. It's very simple in nature. When we look at bacteria, this is what you think of them. This is bacteria growing on culture media. That's kind of gross looking. This is even grosser looking. And it's bacteria growing on culture media. You can see how the other one was kind of shiny. This is all kind of mucoid and slobbery and yuck. Sometimes we see little colonies like this, and these are greatly magnified, and you can see that they get some different formats in them. And they're all bacteria. They're single-celled organisms, and these single-celled organisms have quite a lot of cells in there, but they're all single-celled organisms. And this is a colony that I saw, and this kind of looks like a brain, but it's single-celled organisms, and all they're doing is forming on top of each other. When we look at prokaryotic cells, we often talk about shape, because the shape of the cell makes a difference. We have what we call a coccus, which is round, 1-to-1, and they would look like that. We could have a cocobacillus, twice as long as it is wide, so it's kind of barrel-shaped. We have a bacillus, which is elongate. It's 3-to-1 or larger, so it's more cylindrical, and they would look like that. We can have what we call a vibrio, and a vibrio is kind of a strange shape. A spirillum, which is twisty. It's like a bacillus that's kind of twisted out, and that would look like that. We could have a spirochete, and spirochetes are particularly nasty, and they're like these little filaments, and they would look like this. Now, the length of this is about the same as the length of the bacillus that we've got in the photograph in there. So it's not that they're huge or whatever, but they're very tiny and narrow in their format. Pleomorphic ones have shape that varies. When we look at cellular arrangements, there are 3 planes. You have got the X and Y axis and the Z axis, which goes through the center. If they cleave on one axis, you're going to get new cell. If they cleave on one axis and they kind of stay stuck to each other, we call that a diplococci. If they cleave on one axis and they form chains, we call that a streptococcus. And that's where we get the word strep from. The thing that gives you strep throat actually is a streptococcus. If we get 4 of them stuck together like this, because they divide on 2 planes, we call that a tetrad. If they divide on 3 planes and they stay stuck to each other, then we get that what we call a sarcina, packet of 8. Or if we have them all stuck to each other, but they really don't have any real format to it, we call that a staph, and it would look like this. If it's a bacillus, it's only really going to divide in one axis, but if they stay stuck to each other, we call that a diplobacillus. If we get a chain, we call that a streptobacillus. If we get a chain and it kind of turns into a bunch of columns because they bend on each other, then we would call that a palisade. So there are different arrangements for these cells that we look at. When we talk about bacterial cells, we talk about the type of cell wall it's got. Now up here, it shows you 2 different types of cell wall. The top cell wall is basically cell wall where you've got this thin cell wall on the inside, which is this light blue, and then you've got 2 membranes around the outside of it. And the 2 membranes around the outside of it are shown by these phospholipids in there. And through the membranes, you've got these little proteins, and the little proteins basically allow for exchange of information from one side to the next. If you've got the 2 layers and the part on the inside, that is going to turn out to be a gram-negative type bacterium. If it's got 1 layer and a big thick layer of peptidoglycan on the outside, then we're going to call that a gram-positive. And they stain differently. And the reason we have to know the difference between them is because when we go to treat them, we need to know the difference. The gram staining procedure is rather simple one. It says it takes crystal violet and iodine and alcohol and safranin. And what we do is we start out with a slide. We put the bacterium on it. We put the crystal violet on top of it. That primary stain gets absorbed. Then we add the iodine. The iodine causes the primary stain to get fixed. Then we add the alcohol. The alcohol causes that outer membrane of the gram-negative ones to wash away, which takes the stain away. And then we add a counter stain to it, which is red in color. Red on the purple will stay purple. The red on the clear will make it turn pink or red in color. And that's how we visualize these when we look at them. We can also see structures on the inside of them. These two pictures here are endospores, and endospores are very interesting because they allow for the bacterium to survive long periods of time. And when we say long periods of time, a bacterium in a spore form can survive for tens of millions of years and then come back if it gets the proper conditions. So endospores are very interesting because they allow things to last for a long period of time. And you can see endospores over here as well. They don't pick up the stain when you look at them. These have what we call a capsule. This type of staining is called a negative stain. What it's doing is it's staining the bacterium and staining the background, but it's not staining this gelatinous jello-like stuff around the bacterium. And that's what we want to see in this particular case. And this is called a capsule. And the interesting thing about a capsule is that these tend to be pathogenic to humans and animals. So the capsule is involved in it making you sick. When we look at identifying bacteria, we've got the shape which we can deal with, which isn't all that much because there's only a half a dozen different shapes we can look at. So a lot of times what we do is we inoculate them in media and we look at the reaction you get out of the media. Does it change the color of the media? Does it produce something that we can measure? Does it ferment the media? When it ferments, does it produce gas? Does it break down urea or other materials in there? And when we look at these all become important in identifying it. So when we identify bacteria, we don't always just look at shape. A lot of times we look at what we call here biochemical tests, where they actually break down the material. So we see what happens when they break down the material. So when we look at bacteria are not really complex, but they do a lot of very interesting things.